It's a pleasure to welcome Dr. Sufi Raja, our speaker today from SASTRA University, Department of Biotechnology. Dr. Sufi Raja did his bachelor's from Presidency College, Kolkata in chemistry, and I think he completed his PhD in biochemistry, Department of Biochemistry from Krea University. Then he did one postdoctoral stint in the Institute of Stem Cell Research in NCBS Bangalore, Bangalore Biocluster, and followed by one stay in Germany in Göttingen, the Göttingen University, and where I think we met very briefly because I was living Göttingen when I think Dr. Raja joined Göttingen University. And subsequently he moved to Duke University in USA, and he has joined SASTRA University about 6 months back. And it's a pleasure to welcome him and hear about exploring the coupling between physical properties of mitochondria and its function.
Can you hear me? Nice. Yeah, again, thanks to Kolanda. He's my senior from in bachelor's as well as in Göttingen. We briefly met, as he mentioned. So I think Angelisa will be happy to see us meeting again. So, and thanks for giving me this opportunity to share my research in the field of mitochondria. So today I'll be discussing about how you can monitor the function of this organelle. It's basically an organelle inside our cell. So we it always happens sometimes. Okay. So we hired this organelle. So it became organelle. So it's like billions of years ago, one protobacteria were engulfed by ancient cell, and that eventually became the mitochondria of the modern cell. So for decades we used to think that this organelle is mainly responsible for production of ATP, that is the energy we synthesize in the form of ATP, because to do functions, I mean all the cellular function, you need energy, and that energy is stored in a molecule, special molecule, ATP, and that ATP is being produced efficiently by mitochondria only. But with increasing evidence, we get to know, I mean day by day, that it's not only the ATP production by this organelle, but rather, I mean all the biochemical processes happening in our cell are involved with this, directly or indirectly coupled to this organelle. So because I mean you can do a lot of things. It's responsible for a lot of things like calcium homeostasis you need for muscle contraction and all those things. You have this calcium cycling. I mean that cycling is controlled by mitochondria in one way. And another thing, and that's why it's become very important, that reactive oxygen species. The way ATP being produced in the mitochondria, as a byproduct we produce some reactive oxygen species. In low concentration, it acts as a signaling molecule. That means it helps you to conduct certain biochemical reactions. But if you have more of it, then bad things starts happening. You develop a lot of diseases, and that's why currently, I mean this is a huge circle. Currently you can think of any degenerative diseases. Most importantly, one you heard about is the neurodegenerative diseases like Alzheimer, Parkinson, to some extent Huntington. All those diseases are directly linked to the dysfunction of this mitochondria. I mean if it is working fine, I mean if it doesn't work fine, I mean you will develop lot of diseases. And so increasingly, with increasing evidences, I mean it's becoming I mean, very important to not only understand the function of this organelle and connection between the dysfunction and diseases, but also to design mitochondria targeted specific therapeutic approaches. So, currently the state of the art is to the measure the mitochondrial membrane potential to know the function of the of this organelle. Then what is mitochondrial membrane potential? So, the way mitochondria synthesize ATP is by running a electron transport chain. It's nothing but just you transfer a high energy electron which is contained in NADH. I don't know whether this is working. No, nothing is working. So, in NADH you have some high energy electron, and that is transferred to a terminal electron acceptor, that is oxygen, and in doing so you basically pump some protons from, as you can see from the cartoon diagram, that mitochondria is a double membranous organelle. So, you have an outer membrane, and inner membrane is highly convoluted. It has some invagination, and those invaginated structures are known as cristae. And you have some specialized proteins and protein multimeric protein, basically as self-assembled protein molecules, which form some complexes, and that complexes are basically transferring the electron from the high energy intermediate, that is NADH, to the electron. And by doing so, it transports certain protons from the internal area of mitochondria to the space between two membranes, right? And the important thing is, it is not energetically favorable, because the internal area you have low proton concentration and you have high proton concentration in the space between two membranes. Again, you are transferring some proton from the low concentration regime to high concentration regime. It's an energetically unfavorable reaction, right? Because you need to give some energy to do so. And that's how it develops a proton motive force. It contains one part is the membrane potential and another part is the pH part. Membrane potential, because you are transferring a charged moiety, a positive charge outside of the mitochondria. So, you are basically developing a negative potential, right? And to do so, subsequently you are doing another thing, is like you are changing the proton concentration, which basically pH, right? Proton concentration is pH. So, you are changing the pH of the compartment also. So, you have two components in this proton motive force. One is the membrane potential, another one is the delta pH, the pH difference. This proton motive force basically gives you the driving force to produce the ATP. When it's dissipating through the ATP synthase, which is complex 5 here in the bottom of that cartoon diagram. So, you say it's in the Roman as a V, that complex 5 is basically known as ATP synthase, and that basically dissipates the proton gradient, and in turns it produces some ATP, right. So, all the dye what we have present day we use for measuring the mitochondrial activity is basically measuring the membrane potential, because you develop a negative membrane potential in the inner membrane of the mitochondria, and if you have some positively charged fluorophore, I will come to that what I mean by fluorophore. Basically it will give you some light so that you can see whether your mitochondria is active or not. So, those are known as potentiometric dye, because they are positively charged and you have negative component in the mitochondria, and they will interact and basically localize in the inner membrane and give you some light readout, right. So, this is the current scenario. We use this type of dyes to measure the mitochondrial activity in terms of membrane potential, because the more membrane potential you have, that means more electron transfer is happening, more ATP will be produced, right. And the more membrane potential you have, more dye will accumulate in the membrane because of this charge interaction, and you will have more light readout from the mitochondria. But this linear relationship doesn't hold always, because there are lot of examples where this mitochondrial membrane potential can be misleading. Sometimes it's not sufficient to tell give you an overall picture of the mitochondrial activity, but sometimes it can be misleading. Why? I have listed 3 such scenarios where mitochondrial membrane potential can be misleading. The first one is the row cells, where you don't have any mitochondrial DNA, because mitochondria, I said in the first slide, is like it was bacteria initially and we engulfed them, so still they hold certain amount of DNA inside it, so to synthesize their own protein, right? So there are certain situations where you don't have any mitochondrial DNA. That means mitochondria should not be active, but still you pose some mitochondrial membrane potential in those type of mitochondria. It's not active, but still you have membrane potential. So if you use those commercial dye, you will say that okay, my mitochondrial is active, but it has no component to be active. So this is one example of to be misleading by using this type of potentiometric dye. And the second one is the reverse rotation of the ATP. I said the complex 5, which uses the proton motive force dissipation to synthesize ATP through a rotation. It's a rotor molecule basically. It rotates and transfer the mechanical energy to chemical energy by synthesizing ATP from ADP. But surprisingly, it can rotate in other direction as well. In that condition, it basically uses the ATP to generate the membrane potential, not like it uses the membrane potential to produce ATP. Because unless and until mitochondria is producing ATP, what's the purpose of mitochondria, right? Mitochondria should produce ATP, but in this situation, it will consume the ATP to keep the membrane potential intact. So there is a second example. And third example, sometimes I said this proton motive force, which is basically the main active force to synthesize ATP, has 2 components. One is the potential, another one is the pH difference. Now, in certain scenarios, what can happen is like, even though your membrane potential is getting low, but it can be compensated by delta pH. You can increase the pH difference and compensate for the reduction in potential to keep the proton motive force intact to carry out the ATP synthesis, right? So these are, I have summarized these 3 conditions where membrane potential can be misleading. So day by day, chemists are trying to develop new, smart, robust dyes for measuring the mitochondrial activity in real time in live cells, right? So to address this problem, we thought about addressing some evolutionary conserved processes, right? So there are certain processes where are very conserved throughout the evolution. One such example is the cell volume, because whenever you have increase in cell volume, it's a trigger for cell division. So cell try to conserve the volume by running lot of membrane transporter, channel and all those things, so that you release certain molecules, water and everything to keep the volume conserved. And other such evolutionary conserved phenomena is the homeoviscous adaptation. What is this? It's keeping the membrane fluidity at a constant level, because membrane is the hub for all the most of the important biochemical reaction. So to carry out those biochemical reaction in an optimum rate, you should have a optimum fluidity of the membrane. So cell try to keep that fluidity in an optimal level by changing the composition of different type of lipids. By you have different type of lipids, you can change the fluidity, right? You can have crowding lipid like cholesterol, which can rigidify the membrane, and you can change by having different composition of crowding lipids, you can change the fluidity of the membrane, right? So we thought, what about targeting the mitochondrial membrane fluidity and check what is happening and what is the connection between the activity and membrane fluidity of the mitochondria. So in this talk, I am going to cover these 3 areas basically. How we design a membrane fluidity sensing probe, fluorescent probe, and the coupling between the fluidity of the mitochondrial membrane and mitochondrial activity. And I will give you some example of how the possible physical phenomena that can be coupled or related to, you can use it for changing the fluidity of the mitochondrial membrane. Before I go to the design part, I would like to spend some time with this beautiful phenomena, which is fluorescence. I always love to talk about this because it always amazes me. So you can see it's a cartoon diagram, like you excite the molecule with light, it spends some time in the excited state, then it comes back, right? And when it comes back, it gives you some light that is known as fluorescence. But if you think about, don't go with the absolute time scale, which is like nanosecond, femtoseconds or picoseconds, but think about the time gap. So excitation happening in the 10 to the power minus 15 order of time, and the emission or the coming back phenomena is happening in the 10 to the power minus 9. So you have a 10 to the power 6 order of time difference. Can you imagine like how big it is? Just forget about the seconds. If you replace it with years, so think about 1 million years you are spending in certain stage, right? So in that, that's why in that excited state, many things can happen to that molecule, and finally it decides the fate of that molecule, whether it will be very bright, very fluorescent, or no fluorescent. Everything decided in that excited state. If the excited state molecule loses its energy through hitting other molecule, hitting the solvent molecule, or reacting with other molecule, and it dissipates all the energy, you will don't get any fluorescence from that molecule, because it will go through other non-radiative processes, right? So one such I said the energy dissipating process is basically the intramolecular rotation. If the molecule has some flexibility to rotate along the axis of the molecule, it will do so. And by doing so, you can release some energy, and it can happen that you will not get any fluorescence from that molecule. So we use that trick, like this rotation depends on the viscosity of the solution. If you have very high viscosity, you can stop this rotation and you will get back the fluorescence. But if your solution is less viscous, then it will rotate and you will not get any fluorescence, right? So if you have more viscous solution, you will get fluorescence. If you have less viscous solution, you will get no fluorescence. So that is the idea to develop this type of dye. So we used a simple scheme, microwave based synthesis. We used two molecules and exposed to microwave overnight, and we got this fantastic molecule. And the skeleton is known as hemicyanine based probes, so we call them hemicyanine. And you can see this blue arrow thing along the molecule, along the axis of the molecule, you have certain bonds that are the major sources of rotation. If your solution is less viscous, we confirmed this by doing some quantum chemical calculation, by measuring the potential energy and by changing the dihedral angle or the angle along those particular bonds. And we saw that those are the bonds which are more susceptible to rotation if you have less viscous solution, right? Then the first thing we do when we design or synthesize a new dye, we go for a solvatochromic studies. That is basically checking whether this molecule fluorescence is sensitive to solvent polarity, viscosity, pH, and all those things. So we figured out that this is the quantum yield, that basically a measure to tell you whether how bright is the dye, how good is the as a fluorophore. We measure this quantum yield property. So you can see in the glycerol, for all the dyes, like it's a library, we have extended this library recently by adding more and more moieties to that basic skeleton. But right now I'm showing you only 4 dyes. So all of them have one common property, is the increase in fluorescence intensity is the high viscous solution, that is glycerol. Glycerol is 1000 times more viscous than water. You can imagine like how viscous it is. If you have worked with the glycerol, if you want to pipette it, your life will be like miserable, because it's not easy to pipette glycerol because it's so thick, so dense. So it's a clear evidence that if you have high viscous solution, your fluorescence property will increase. So we checked this more systematic way by changing the viscosity of the solution by adding increasing amount of the glycerol, and you can see the fluorescence intensity is gradually increasing with increasing viscosity of the solution. And the beautiful part of this dye is the fluorescence lifetime also increases with increasing viscosity, and that is more important because this is the intrinsic property of the fluorophore. It doesn't depend on the concentration of the dye. I will cover this, why it is so important. You can do quantitative analysis using this lifetime. The lifetime and fluorescence lifetime is, I showed you in the that excitation emission plot, that the excited molecule spends some time in the excited state. That you can basically measure, like how much time is spending in that excited state, and that is known as lifetime, fluorescence lifetime. And that lifetime is also increasing, because by increasing the viscosity of the solution, you are keeping them in the excited state for the long time, so that it gets time to fluoresce, right? And after checking this in the solution mode, we asked whether we can sense the viscosity of the membrane. So we used a model membrane, like we made some vesicles with increasing viscosity by adding some cholesterol and sphingomyelin. Those are some complex lipid that basically increases the viscosity of the membrane. So you can clearly see the lifetime. We did some fluorescence lifetime imaging here, and you can clearly see that with increasing amount of the cholesterol or the sphingomyelin, which basically causes rigidity in the membrane, your lifetime is also increasing. So yes, our dye is sensitive to the membrane viscosity as well. But the next question, whether they are sensitive to the mitochondria or not. So we did one co-localization assay. Here you can check the localization by having, in one window you have a mitochondria specific known dye, and another window you can do two color imaging, one for the specific dye which is known, I mean everybody knows, and another with your own synthesized dye. And when you overlap them, if they overlap perfectly, then we say that's okay, it's going to the mitochondria. One way of doing that, I mean the standard assay. But I said that mitochondria have two membrane, right? Then which membrane? So then we did the super resolution microscopy, that is STED. So from Göttingen as well. Stephen Hell got Nobel Prize in 2014 for this microscopic technique. So you can have more and more resolution. So from this STED image you can see the dye is staining those invaginated structure inside the mitochondria. From this we can conclude that our dye is highly specific to the inner membrane of the mitochondria, not the outer membrane. And before I go to the next question, so we have the dye for sensing the mitochondrial membrane viscosity, but what does it mean? What is the connection between mitochondrial viscosity and the membrane and the activity of the mitochondria? Before I go to that part, I want to do some advertisement for our dye, because it's so good and I love this dye. So one thing is, if you use the standard commercial dyes, which are basically, one of them is the TMRE, I mean its full form is the tetramethyl rhodamine ester. So all the commercial dye are mainly rhodamine based. You can see inside the cell, these are some primary neurons, those are sensitive cells. In the middle of the cell, which is basically the cell body, you can see some background fluorescence is coming up after a certain time of imaging. That basically happening, you are introducing some phototoxicity. By introducing phototoxicity, you can reduce the membrane potential. You are basically damaging the mitochondria. And if you reduce the membrane potential, your dye will come out of the mitochondria. So it will start giving you some fluorescence from the cell body. That's why you are seeing some background fluorescence. It's saying that you are basically damaging your mitochondria, but no such thing you can see from the synthesized, the dye we synthesized. And the interesting thing is, though we used more laser power, you can see that 10 microwatt for the commercial dye and 15 microwatt for our dye, but still we didn't see any measurable toxicity in terms of photo toxicity. Then another few desirable properties to do mitochondrial imaging. The lower, it's a long-term imaging. I did it for like 1 day, 24 hours. So you can see it's these are migrating fibroblast. I imaged them for overnight, and you can see there is no cell death or cytotoxic effect or any fragmentation of the mitochondria. That's a signature. If you have more and more fragmentation, that means your mitochondria is getting damaged. And there are other certain things we did, like to check the cytotoxicity. There's a standard assay, MTT assay. You can use that to see like whether the cell is dying or not. So we used higher concentration than we use for imaging, but still we didn't see any noticeable cell death by this type of dye. So let's come back to the question, what is the connection between mitochondrial membrane fluidity and its activity. So this is the cartoon diagram of the reaction scheme when you transfer the electron from a high energy intermediate to the terminal electron acceptor, that is oxygen. So in between, I mean this transfer is happening by some complexes. There are 4 complexes, 1, 2, 3, 4. So this is a complex arrangement, but electron transfer happening through these complexes. But in between, you have some mobile carriers. This UQ you can see, those are basically quinone. It a mobile electron transporter, and another transporter is the mobile carrier. Another one is the cytochrome C. So these are 2 mobile carriers. We call it the mobile carriers because it's hop from this carrier hops from one complex to the another complex and carries the electron from one complex to another complex. So you can imagine that to have certain level of to transfer or to make them active in a rigid membrane environment, you need to have some flexibility, right? So it's obvious to think that more fluidity means more transfer of the electron or the movement of this type of mobile carrier. So there should be some connection between the diffusion properties of this or the diffusion behavior of this mobile carrier and the membrane fluidity. To check this hypothesis, we used this fluorescence lifetime imaging, and this basically it's a quantitative imaging. Why I always I'm saying about it's a quantitative imaging, because intensity based imaging depends on the dye uptake. The more dye you have in the mitochondria, the more intensity you will get, and that can be sometime misleading, because if your one plate have you have like 100 cells, in another plate you have 50 cells, and you used same amount of dye, so the 100 cell containing petri dish will give you less fluorescence compared to the 50 cells, right? Because effective dye concentration in the other way, the 50 cells will have more effective dye concentration, then that plate will give you more fluorescence. But this change in fluorescence is an artifact, because your effective concentration is varying depending on the cell number. But if you do fluorescence lifetime imaging, that is an intrinsic property, it does not depend on the concentration of the fluorophore. So you can do in a more quantitative way. You do not have to worry about how much dye has been uptaken by the mitochondria and all those things. So before I go to the assays, this is one beautiful thing I want to discuss, is like, I mean, in a plate, if you have 100 cells, I mean, it's quite obvious that all the cell will not be in the same energetic states, right? In the same state, they should have some heterogeneity. But when we did like single cell imaging, in one single cell itself, you have heterogeneous environment in the mitochondria in terms of the fluidity, because I said that fluorescence lifetime is directly proportional to the fluidity. I mean, inversely proportional. If you have more fluidity, your lifetime will be less, right? So in single cell, you have different mitochondria in terms of fluidity. And another surprising thing is that even in a single mitochondrion, you have different patches of areas in one single mitochondrion having different fluidity. So this tells a lot of things I will discuss later, but first let me show you 2 assays by which we confirmed the connection between the activity of the mitochondria and the fluidity of the membrane, mitochondrial membrane. So we took advantage of 2 known things to do so. Another one thing is, if you uncouple the electron transport chain with ATP production thing. So somehow if you dissipate the protons from the space between 2 membranes, as per the La Chatelier principle, you will ramp up the electron transport chain reactions. You can dissipate in an ordered fashion, you can dissipate the proton motive force by synthesizing ATP, right? But if you don't synthesize ATP, just use certain transporter or uncoupler or some channel which can dissipate the proton in other way, you are basically increasing the right hand reaction, so your left hand reaction will also increase, that is the electron transport chain, and it is known as basically mitochondrial respiration. So you can increase the respiration by uncoupling the electron transport chain with oxidative phosphorylation or the ATP production. And another thing we took advantage of is the primary source of the electron, that is NADH and FADH, they are autofluorescent. That means those molecule itself gives you some fluorescence. You don't need some fluorophore to check the level of this fluorescence of these metabolites, NADH and FADH. They itself give you some fluorescence. So we use that thing and figure out and did some assays like the connection between the autofluorescence of these molecules and membrane fluidity. How we did this? We did, two channel imaging. In one channel, we measured the fluidity in terms of fluorescence lifetime imaging, and in another channel, you have this film. So we measured the autofluorescence lifetime of the NADH itself. So it's known in the literature that and the field as well, the more respiration you have, the lifetime of the NADH will also increase. So when NADH is in bound form means it's associated with the electron transport chain complex, it will give you more lifetime compared to when it is in the solution or the mobile condition. So you can see from this histogram diagram, the red dotted plot is in presence of the FCCP. FCCP, I forgot to mention, is basically uncoupler. I said about the uncoupler, you can uncouple the electron transport chain with the ATP production. So we use this chemical to uncouple these two process and to ramp up the respiration. So you can see respiration has been ramped up by using FCCP because your histogram has been shifted to the right side. And it's known in the literature, it's a standard assay. But interestingly, when we compared or correlated this with the dye lifetime, we observed a negative correlation. Quite expected, our assumption was like that, right? So you need more fluid membrane to carry out this electron transport chain, right? So it's this negative correlation is saying that same thing. When you have more lifetime for the NADH, that means you have more respiration, but at the same time you have less lifetime for the dye. That means membrane is more fluid when your respiration has been ramped up by the uncoupling the ATP production and the respiration. But it's a kind of brute force because FCCP, if you use uncoupler, in long run, you are basically killing or damaging the mitochondria. So we thought about another standard assay. This is being used by many other, people like Jennifer Lippincott-Schwartz, who is known in this mitochondria field. So if you starve the cell, means if you don't give glucose. So if you are culturing cells in vitro, you need to give some food to them, right? So if you don't give that food like glucose in the media, basically you are making them to starve. So if you do that, it will start using its storage, mainly the lipid storage. And it is known that when it does so, the cell does so, it basically uses the mitochondrial ATP production. It is known. So we thought about checking our dye lifetime in those contexts. Here also you can see in the histogram is clear, and as well as those scattered plot is clear that in the red it's basically showing you the fluidity or the fluorescence lifetime of the dye in control cells, but when you starve them, the fluidity is increasing because your lifetime is decreasing. Again, I am saying it's a inversely correlated relation, right? Dye lifetime and fluidity. So it's a clear evidence that yes, actually, you need to have. So this is our proposed model. You need to have fluid membrane in the mitochondria to carry out all these electron transfer reaction to ramp up the respiration and thus to increase the mitochondrial activity in terms of ATP production. So it's a close relation between mitochondrial membrane fluidity and eventually the ATP production. So one can use this probe to measure the mitochondrial fluidity and have some idea about the activity of that particular mitochondria. And
how do we understand the first graph?
sorry, sir.
First set. So starvation control.
Yes.
So yeah, it's a distribution of population in various lifetimes.
it's each points are a single cell.
Correct. So it corresponds to one lifetime, like 800.
Yes, average lifetime of that particular cell. So next, to understand what are the possible mechanism behind changing mitochondrial membrane fluidity. So one can think about in one direction. Obviously, one direction is like to check what type of lipid you have in different location of the membrane, and if you are changing the lipid composition in some way to change the fluidity of the membrane. But the time scale of these changes, like if you start the cell within 10 minutes, you start seeing the changes in the fluidity of the membrane. So you can have some lipid vesicle fusion to the existing membrane. It also happens in faster way. So one way of thinking or addressing this issue by studying the lipid composition and how if lipid fusion is happening in that time scale or not. So for that I am doing some with my previous postdoc, I am having some collaboration to understand the molecular mechanism, like what lipid pathways are involved in controlling the membrane fluidity. But being in a physics lab in second postdoc in Duke, I started asking some question about the physical basis. So if you can think about, suppose you are, how you can change the fluidity or the flexibility inside a membrane. If you stretch a membrane, definitely you are increasing the tension of the membrane, but you are increasing the inner volume, like void volume. The interaction between molecules is losing up because you are stretching the membrane. So you have more void space between two molecules, right? So you can basically increase the diffusion properties inside those membrane if you stretch it. And this type of thing you can do in two ways. One, it's stretching this membrane with some anchoring point, you are stretching it, or you can increase the volume of the mitochondria. If you increase the volume of the mitochondria, you can also increase the tension of the membrane, right? Suppose you have a membrane highly invaginated and you are swelling the mitochondria, or you are increasing the volume, inner volume, you are basically increasing the tension of the membrane. Basically, you are increasing the the void volume inside the membrane. So to check that, the first thing, like whether when mitochondria is active, your volume is also increasing or not. So checking the volume is really difficult in present days microscopic technique, optical technique, because using STAID you can reach, like theoretically, like your resolution can reach like 10 nanometer or so, but still your axial resolution is limited. Axial resolution means this height, right? So height resolution is still poor. You cannot go below that. So and the problem is the distance between two membranes. The one way of measuring the volume is you can measure the distance between two membranes. If you can see that the distance between two membranes is decreasing, the average distance, that in that way you can also say that your volume is increasing because the distance is decreasing. But how to check this whether the distance is decreasing or not? Because if you see in the EM image, this one, the distance between two membranes is roughly 10 nanometer. So this 22 nanometer is basically including those two membranes. So one membrane has a thickness around 6 to 7 nanometers, right? So if you subtract that amount, so the distance is basically around 10 nanometer, and which is beyond any existing optical scope of any optical microscope. So in collaboration with Jörg Enderlein in Göttingen, they developed a microscopic technique by which you can basically measure the sub-nanometer resolution. The distance of, you can basically study one single leaflet of the membrane. So you can go below 1 nanometer. The principle is very simple. So if you have a quenching surface means it can quench the excited molecule. It can reduce the fluorescence. And this reduction of fluorescence depends on the distance from this quenching surface. So the more closer you are to the surface, the more quenching will happen. So you can reconstruct a map, height map, depending on the quenching. right? So in that context, what we used a single layer of graphene, which is known for quenching the fluorescence. Due to some near field coupling, the excited electrons will be transferred to this graphene sheet. So if you we use this graphene sheet and place some isolated mitochondria and checked, the reduction in the fluorescence property in inactive state and active state. So in active state, we found basically there is a reduction of the average distance between the two membranes reduces up to like 2 nanometer. So it's a clear evidence that basically when your mitochondria is active, your volume is increasing. That means you are creating some tension in the membrane. You are basically stretching the membrane in a by increasing the inner volume of the mitochondria. And it's known how you can do that, because when you are when the mitochondria is active, you need to have a lot of things get inside. Like you need ADP to synthesize the ATP, you need other ions also. So you have many channels, and you have channels for water transport also when ADP is getting inside and other ions are getting inside. So you have a little bit swelling of the mitochondria when it is active. So it makes sense, like you can increase, basically your mitochondrial volume is increasing when it is active, and we can probe that by using this type of state of the art super resolution technique. And another way, like I discussed, like if you pull the membrane, so how you can do that? If you this is one thing, that's why the idea came to my mind. These green spots are basically mitochondrial DNA. So I said that mitochondria were bacteria, and bacteria have their own DNA. So mitochondria also, they have some DNA, and those DNA are basically not like nuclear DNA, long chain-like thing. Rather, they form a spherical circular DNA by the help of some transcription factor, and those condensates are known as liquid droplets, because these are flexible things. Sometimes they during protein synthesis or replication, DNA replication, it swells a bit, but in general they form a droplet kind of structure. And the dimension of this droplet is around 100 to 120 nanometer. And the thickness of the mitochondria, you can see, these are the isolated mitochondria, that's why it looks big. But in the elongated mitochondria in the cell, the distance is around 300 to 400 nanometers. So you can think of like what will happen if this mitochondrial DNA, and it's known to be connected with the membrane, is moving and changing that, is dragging the mitochondrial membrane in different direction and changing the tension of the mitochondrial membrane. I will show you one beautiful example, beautiful video. Whatever I said, this is the linear mitochondrial DNA, and they form condensate by the use of some transcription factor, and that's how you see them in a spherical form in the real life. And in confocal microscope, it's not a confocal, it's a semi super resolution AD scanning microscope. So you can see these green dots are basically mitochondrial DNA, and the red is mitochondria. And you can see lots of, in some cells you can have like close to 1,000 of these mitochondrial DNA spots in one single cell. So this is the thing. You can see within a single mitochondrion, these green spots, the DNA are moving. And if they are attached to the membrane, you can, it's quite normal to think that your membrane will be dragging by this type of movement, right? So the first thing we wanted to understand, what is the type of this movement? Like, is it diffusive? It is thermally driven, or is there any active player are there to make this movement happen, right? So, but there is a intrinsic problem to it, like in the context of analysis, because you can see your backbone is also moving. It's not stationary. So you need to have a reference point, and so to make things simple, what we did, we basically measured the fluctuation between distance between two spots. If you measure the fluctuation of that thing, in some way you can get rid of the global fluctuation, right? So we measured the distance, and we plotted the mean square displacement of this type of fluctuation. ATP from the cell, there is no fluctuation, but there is a diffusive fluctuation, can be probed by plotting the MSDs like mean square displacement. It clearly says that this movement is active in nature, it is not thermally driven. And then the next question we asked, if it is active, then who is driving this process, right? So the known player to drive active process in the cell, either microtubule or cytoskeleton, your actin or the myosin, non-muscle myosin, they are contractile in nature, they pull things, right? So we blocked the myosin activity by your blebistatin, this PNB, paranitroplebistatin. Then nocodazole we used to block destroy the microtubule, and cytocollagen B to destroy the actin network. But surprisingly, nothing happened to this movement. So it's not thermally driven, and it's not dependent on any of the known cytoskeletal component. Then who is driving this? That is the main yes. It's highly heterogeneous, and we are trying to refine it in terms of absolute thing. That's why it's an.
Actually, conclude there is a rising trend.
Sorry, sir.
The kind of error bars that you have, you seem to have concluded that there is a rising trend as time goes up, right?
Yes. The mean has a rising trend basically. Yes. Oh, yeah, sure. Because if I think about this, all this, what I say, this G prime, G double prime, viscoelastic response thing. I mean, there you have a lot of error thing. But this is definitely, the error bar is really huge. But we are considering on the mean thing, and as I said, to make things simple, I used the analysis of the fluctuation between two dots, the distance between two dots, right? To make it absolute, I'm basically working with one guy in Göttingen, so to use the AI and to measure the absolute fluctuation, not like these are some qualitative analysis, right? So these are my future plan regarding this context. To find out the source of this type of fluctuation, what are the players, if there is any contractile machineries inside the mitochondria, and if the crystal dynamics and this type of dynamics is coupled, and if so, then how it is coupled with the mitochondrial membrane activity, so that we can correlate this tension thing, like your mitochondrial DNA is dragging those membrane, and you are basically changing the activity in a dynamic fashion. But as I said, to make it more, to find out the connection between crystal dynamics and your and activity and motion of the mitochondrial DNA, you need to have high resolution imaging. But this is the problem of the high resolution imaging. Super resolution imaging, 1 or 2 snaps is good, it's fine. But if you want to study the dynamics, you will introduce toxicity, because it's a very you need very high power laser, and those lasers are very bad for life systems. You can see mitochondria swelling and finally rounding up. That is a clear signature of mitophagy, means the mitochondria is going to die, right? You are doing this, you are having this toxicity because of the art of the imaging. You cannot avoid these things. So to address these things, you can use deep learning based technique, like you can the same component, you can have 2 images, one is the low resolution, one is high resolution, and you can feed the system with the information with the high resolution image, so that you can extract the same information from your low resolution images, right? So this type of thing you can do, and I'm collaborating with Daniel for this, not only for this image processing, also for tracking those things using AI. And apart from this, my main future research focus is to understand the mitochondrial heterogeneity in the context of cell function. So I showed this image before, that inside a cell, all the mitochondria are not in the same state, right? Why? So that's what I want to understand, like whether cell itself has an active management system to control the activity of the mitochondria so that it can provide the energy where it is needed at right time, at right place. Suppose I'm giving you one example. During cell migration, your cell is migrating. We still don't know whether your leading age, like the frontier age, the mitochondria in the frontier age are more functional compared to the lagging age. So those type of things I want to understand, and to understand if so, then what are the possible mechanism could be, like during the migration is really an energy consuming process, or also lot of signaling event involved, like row, rack and all those signaling events. So am I on time, or do I? 5 minutes is fine. 5 minutes I can finish it. Yeah. So yes. So to check, like if the activity in different location of the cell is different depending on the cell function or not, and if so, like how you can control those type of heterogeneity in the space and time inside the cell. So I want to understand also the connection between signaling dynamics in the context of migration and the mitochondrial dynamics. So for this, I've recently started a collaboration with Andre in UIC. He is an expert in developing optogenetic tool. So using light, you can activate certain proteins in anywhere of the cell where you want. Like if you shine some light at that portion, your certain proteins will be activated. So I want to see like how during the migration, in the context of migration, if I use activate certain signaling cascades relevant to migration, and how mitochondria is responding to those type of activation. With this, I think this is my, yeah. Okay, then I'm on time. So with this, in summary, I showed you that we have developed some mitochondria membrane fluidity sensing dye, fluorescent dye, and I showed you that this fluidity of the membrane is tightly coupled with the mitochondrial function, and using a state-of-the-art microscopic technique, I showed you that distance between two membranes basically reduces when you activate the mitochondria, and I also showed you like that mitochondrial DNA you can use to probe the viscoelastic properties or the diffusion properties inside the mitochondria, and this type of diffusion is basically active in nature. And with this, I would like to acknowledge my PhD supervisor and all my postdoc advisor and all my lab members in those labs, and these funding agencies for supporting me. And with this, I stop here.